Good afternoon, everyone. I guess we should go ahead. On behalf of the University of Peradeniya and Faculty of Agriculture, I welcome you all to the Agricultural Biotechnology Center and to this special seminar on nanopore sequencing. I especially welcome our speaker, Dr. Lakmal Jayasinghe, here with us today at the Agricultural Biotechnology Center. Before I introduce the speaker, let me brief some logistics. His talk will be about 40 minutes, and then we will have a Q&A session. Please type your questions in the chat area just to save time. And if time permits, we can have a discussion as well. Until others join, let me begin with my experience. I was a big fan of Illumina sequencing since its inception. Even though there was no service provider in Sri Lanka, I have been using the technology for many years. I mean, Illumina. As many of you know, there's a lengthy procedure to send samples to foreign countries from Sri Lanka. There's no reliable service provider, at least for Sanger sequencing in Sri Lanka, as many of you know as well. I had a dream of establishing a reliable sequencing service in Sri Lanka. However, Illumina machines are very expensive and they keep updating quicker than we think of. Therefore, we have been sending the samples to the USA and Korea for our work. It was very tough sending RNA samples for RNA-seq studies because we have to send all our samples door to door in dry ice. I learned about this new technology, Nanopore, around, I guess, 2016, 17 or so. However, I did not have any idea moving from Illumina. One step forward, Agricultural Biotechnology Center established a bioinformatics facility with funding received from a competitive research grant from the National Science Foundation to work on cinnamon. After receiving funding from the DWC to work on Department of Wildlife Conservation, to work on several other genomics projects, I hired several computer engineering students to work on our data analysis. So our bioinformatics facility is a collaborative facility among the members from the Faculty of Engineering, University of Peradeniya, and the Agricultural Biotechnology Center. One of them hired Hiruna Samarakon. His undergraduate project was on Nanopore sequencing platform. It was again a collaborative approach. Every day he was telling me, "Madam, why don't we establish our own system?" He was bugging me, basically. We should establish our own system. I'm not sure whether he's in the audience. He's now in Australia. He convinced me to read more about nanopore sequencing and linked me to Dr. Lakmal Jayasinghe. Apparently, Lakmal and I have a common friend, one of his batchmates in Davis, California. Even though we started this collaboration, things were moving a little bit slower. However, the COVID-19 accelerated everything. As you know, it changed many things as well. We could not send samples because of lockdowns, and all the projects got delayed. Then I requested funding bodies to allow us to utilize the funds allocated for Illumina sequencing for establishing our own nanopore facility. I should appreciate following funding bodies, our funding bodies actually at this point. OWSD, Organization for Women in Developing Countries, it's a UN organization. So I have a research grant, a fellowship. And then the National Research Council of Sri Lanka. So they approved our request, and all necessary equipment for the quality testing and this tiny machine that Lakmal is going to present will be provided or will be gathered through this funding. So I want to acknowledge them as well. Since Dr. Lakmal visited at the same time for a visit from London, I thought it would be a good idea to introduce nanopore sequencing to our scientists. Here we are. So that's the journey. I wanted to tell you how we established this collaboration and where we are now. Let me introduce him formally now. A short bio. Dr. Lakmal Jayasinghe is the Vice President of the Research and Development of Biologics at Oxford Nanopore Technologies. He studied at R&D College, Colombo, and read a chemistry special degree in the University of Colombo before he completing his PhD in chemical biology in the University of Oxford. Lakmal joined Oxford Nanopore in 2006 as the first lab scientist and kick-started biological research in the company. During his PhD in the Hagen-Bailey group, Lakmal has studied different nanopores and has gained a wealth of knowledge in engineering proteins using genetics and chemical modification approaches. His responsibilities at ONT, Oxford Nanopore Technologies, include improving readout signal of ONT chemistry by upgrading his current biological components and discovering and engineering new proteins to suit upcoming ONT applications and platforms. Lakmal also works with many academic collaborators across the world to make sure Oxford Nanopore stays ahead of the competition, of course. He's a proud Sri Lankan, and we are very proud of you, Lakmal, and we are really nice to have you here with us at the Agricultural Biotechnology Center. So with that, I hand over the virtual group or platform for you to talk about your own technology. Welcome, and over to you.
Can everyone see my screen?
Not yet. Let me see. Yes.
Let me go back, share screen. Is that all right?
Yes, we can see.
Okay. So, so thank you very much for the introduction, and it's a pleasure to be here. So I wasn't actually quite sure what the audience and what their expectations are. So I try to keep it like a bit general with enough, hopefully, scientific background. But my idea here is to introduce nanopore sequencing and to show what it can do in a country like Sri Lanka. So a little bit background about Oxford Nanopore and Oxford. So this is the Oxford University. It's a nice campus, and this is the Oxford Nanopore Technologies headquarters, which is located in the Science Park of within Oxford City. So the company was founded in 2005 as a spin-out company from the University of Oxford, founded by Hagen Bailey, who is my PhD professor. So at the moment, we have about 800 people, and it's a very kind of interdisciplinary company with scientists from biochemistry, molecular biology, bioinformatics, engineers, manufacturing, commercial people. We have labs and offices all across the world now, including New York, Boston, San Francisco, Japan, and many places in China. And a couple of months ago, we did an IPO, a public offering. It's one of the most successful biotech IPO in London Stock Exchange, and currently the company is valued over 7.5 billion US dollars. Not working, hold on. Yeah, okay. So this is the MinION, which is our most popular system, handheld DNA sequencer, and behind that is our manufacturing facility in Oxford. So we have a very global long-term IP strategy as well. So everything we do, we file IP. We have more than 1,500 issued patents in approximately 200 patent families. We have a huge R&D department in biochemistry, molecular biology, bioinformatics, engineering, platform development, and all sorts of areas. And research funding, we continue to collaborate with academic institutions as well, and we have a lot of collaborations across the world in many universities. So these are some of the universities that we have partnerships with. So DNA, as everyone knows, is a source code to build and operate organisms, whether it is in metagenomics, in virus, bacteria, mammal, human, or a crop. By understanding DNA, you can answer many questions. You can understand what is it, what's in it, and if it is a virus or bacteria, whether it is changing and responding to vaccines or any other medications. Is it healthy, diseased, and so on. So in plants and metagenomics, and you can also understand how the environment change the DNA as well. So understanding DNA is important, and when the human genome was first originated, it took about 13 years for a team of international scientists to complete the human genome. Cost about $13 billion US, but over the years, the cost came down significantly. And also the time it takes to sequence DNA came down significantly. So there's a big drive to improve DNA sequencing technologies. There was a program from NIH to 1,000 Genome Project, which is to cut cost to $1,000 to do a human genome. So we are almost there now, and also the time it takes is hours and days rather than 13 years. So there are many different DNA sequencing technologies available. So the oldest methods, people still use Sanger sequencing, and then there were other technologies over the years, first generation sequencing, second generation sequencing, third generation sequencing, and so on. So the Nanopro sequencing is the next generation sequencing company. So we are one of the latest technologies, and some of the other competitors are like Illumina, PacBio, BGI, and so on. So at least by the time when we started DNA sequencing, Oxford Nanopore Technologies, most of the other DNA sequencing technologies were stuck in this era we call a mainframe stage. So if you take computing, there was this mainframe stage, which turned to, then came the desktops, the mobiles, and wearable sensors, and so on. So in terms of sequencing technologies, most of the companies had these big, bulky, floor standing machines. By the time we started DNA sequencing, and then at the moment, actually, some of the other companies also have smaller desktop machines, but we started these portable sequencing devices. So our goal is to enable the analysis of anything by anyone, anywhere. And to do that, you have to make a portable DNA sequencing systems. Our idea is to connect all the dots, whether it is in health or research, in security, population genomics, industry, education. There are many different areas that you can use sequencing technologies. And to access all these areas and to connect all these dots, you need to be able to provide reliable, scalable, and affordable sequencing technologies. So let me now go through the, as an introduction to Oxford Nanopore technology. So it's based on something called Nanopro Sensing. So the Nanopore is a tiny, is a protein with a tiny hole. It makes the hole in a membrane. And then what we do is we pass the pass current through this pore. So you can measure that current, as you can see here. And if there is something, if you put something into the pore and it blocks the current or it blocks the ion passing through, and you can see this blocking, and you can measure this dwell time, the amplitude of change, and so on. So depending on the size of the molecule inside the this hole inside the nanopore, and the properties of the molecule, whether it is charged or size and so on, you will see different types of blocking. So that block, we can then bioinformatically deconvolute the signal to understand what it is. So it's like if you take a water pipe, and if the pipe gets blocked, then the water, the flow of water get disturbed. It's sort of similar principle. And then we use this DNA, this sensing, Nanopro Sensing technology for DNA sequencing. So what we do here is, so we have the membrane, and this membrane is synthetic copolymeric membrane, which is very stable. And then we embedded in the membrane is this nanopore, which is the blue part, and this is a protein pore. I can talk a little bit more about the nanopore. And then we pass the DNA through the pore under potential. So DNA is obviously negatively charged. So like if you put positive potential underneath, so it will pull the DNA through. And then we use a motor enzyme to control the movement of the DNA. If you don't use a motor enzyme, it travels through very, very fast, hundreds of thousands of bases per second. But to get enough resolution, we use a motor protein, and it controls the movement. And at the moment, we are running about 450 bases per second. And then you have obviously run conditions, salt, pH, temperature, and so on. So you manipulate all these things to get the maximum resolution. And then you have enzyme, well, magnesium, cofactors, and so on to add to the system, to hold the system. So this is our basic setup. So we have this double-stranded DNA coming through, and the motor protein separate the two strands, and it let one strand pass through, and we read that strand. So when the DNA strand passes through the nanopore, it obviously changes the ionic current. And then depending on the sequence within the pore at any given time, whether it is ATCG, it's not just like one base at a time, but the combination of bases gives a specific signal, and then we deconvolute that signal to understand the sequence. So, and then we make chips, what we call flow cells, and a lot of electronics. And in this, what I'm showing here is, so we have a nanopore array, individually addressable electronics, and then depending on the platform, we have different chips or different flow cells. In some flow cells, we have 126 of these pores in one flow cell. In the mid-ion one, we have 512, and in the largest one, we have 3,000. So, which means that any given time, you will have multiplex system that you can analyze DNA through. So we have to do the nanopore data acquisition, you have to do the sample preparation. So whether it is coming from a DNA, so it's a blood or saliva, or a culture, whatever it is, and you do a sample preparation, and then you use those prepared sample in one of the platforms. Then the DNA is passing through the array, and then we get the sequence deconvoluted into the signal deconvoluted into the DNA sequence using bioinformatics tools. So these are some of the sample preparation. You can either do, we have an automated system, but we also have a lot of kits for many different applications. These are the manual kits, but it's absolutely fine. So people can use whatever they want, and depending on their circumstances. So when our system is real-time data analysis, so by while the system is running, it will analyze data in real time, and it will give you answers. So you will know what is in the sample, and so you don't have to wait until the experiment is completed to get the answer. So that is very important, especially in COVID sequencing and applications like that. So as a result, this is the only sequencing technology that can have this kind of real-time data analysis in a portable system, which is very useful for many applications. So these are some of our platforms. So the Min9 is the handheld device, which is quite handy. And so we have the new version called Min9 Mark 1C. The Min9 doesn't have the computing power, so you have to connect that to a computer or a laptop. But the Min9 Mark 1C has its own computing, so you don't need a computer. And then the grid ion is sort of like 5 Min9s together, so you have enough computing power in the system to run all data analysis. And Promethean is a large system, and it can generate terabytes of data. And the SmidGiant is still under development in R&D. It's a small a mobile phone compatible, a smartphone compatible sequencing system. So just to give rough an idea about the size, so this is the Min9 is the handheld device, and it's very small, very portable. Even the large versions are smaller than some of the other sequencing technologies machines. So chemistry and accuracy. So when you talk about accuracy of a sequencing system, especially from nanopore sequencing point of view, there are certain things that you can manipulate or improve to improve the accuracy. So the sequencing chemistry, which is the nanopore I talked about, and also the enzyme. So we do quite a lot of manipulations. We engineer the two proteins quite a lot. And then we can optimize accuracy by changing run conditions, whether it is the temperature or the voltage and so on. And depending on the platform, electronics and hardware, software, it changes the signal. And then how you analyze data, and when you have the raw signal, when you convert it to the sequence of the DNA. So there is a lot of informatics involved, obviously. So all these things can improve accuracy. So to give a little bit more details about the protein side. So these are some of the kind of motor proteins, which are the enzymes. So DNA, if you don't have an enzyme to control the movement, as I said earlier, it translocates very fast. So what enzyme does is control the DNA movement, so that we can get enough resolution to convert the signal into the DNA sequence. So we have used hundreds of different families of enzymes, polymerases, helicases, and so on. And with each one, we make thousands and thousands of mutants, and we re-engineer them and we rediscover them. And we work with quite a lot of academic collaborators on enzymes as well. It's the same thing with the nanopore. So we have looked at hundreds of different pore families and different shapes and sizes, as you can see. Some are publicly available crystal structures, but if we don't have the crystal structure or the cryo-EM structure, we obtain the structure either directly working with companies or working with other academic collaborators. We have tested thousands and thousands of mutants, variations in each family. So our current nanopore is protein called CSDG from E. coli. So it's a monomeric, which means 9 subunits protein, with a 36 stranded beta barrel. The dimensions and the position of the constriction, which reads the DNA matters in sequencing. So this is kind of like an ideal architecture when we saw the protein. So we have licensed protein from lab in VIB in Brussels, hand rheumat lab, and we are working very closely with these labs to improve the proteins even further. So as I said, we make thousands and thousands of mutants, and at each stage, also like we try to get structures to make sure that we have not engineered it completely out of the structure. So to make sure that we are doing the right thing. By improving these posts, you can improve the resolution. I mean, just to compare one of the previous posts versus one of our current posts, which is R9 series, you can see the resolution between ACGT, and it's better. If the resolution is better, obviously the accuracy will be better. So this is a non-stop exercise. So we keep looking for new proteins, keep improving our current proteins for various applications. So when you talk about DNA sequencing and accuracy, one of the important thing of nanopore sequencing is also the length of the DNA that you can sequence. In some of the other methods, the length is limited, like 400 bases per second, where they are kind of short read technologies. But in nanopore, there is no limit to the length of the DNA that you sequence. Whatever you can put in will be read. I think the current record is 4.2 million bases in one strand, and people do get quite long strands. So when you have a longer DNA pieces, it's much easier to assemble these things together to get the overall picture rather than having small pieces. For example, if you take E. coli genome, which is 4.6 million bases, and if you cut them into 50 base reads, there will be 92,000 pieces. And if you cut them into 500,000 bases, there will be only 9 pieces. So it's much easier to assemble 9 pieces and get the bigger picture rather than trying to assemble 92,000 pieces. So that's kind of the relativity of the DNA length and the resolution. And also, if the length is longer, it's easier to get the consensus accuracy. So consensus is, so so you have this long DNA strand, so you can pass them through the nanopore and then you can get the accuracy. So we call that the single read accuracy or the simplex accuracy. And then you can pile up these simplex reads, and then you can get the consensus. So the how many reads that you need to pile up to get enough consensus is the depth, what we call. And then depending on your requirements, you can add more single reads to get a reasonable consensus. So in nanopore sequencing, one of the drawbacks, like people talk about when talk about nanopore sequencing, is they say its accuracy is lower than some of the other competitors, but our accuracy is actually improving significantly. It's over the years, it has improved a lot. So at the moment, it is around, so we have a few different chemistries. The chemistry that most people use at the moment is called this R9 chemistry, which means the pore is called actually R9. And with that, we are hitting about Q18 accuracy. So if you take Q score, Q20 is 99% accuracy, 1 in 100 errors. Q30 is 99.9 accuracy, 1 in 1,000 errors. So that's where we are moving. And this is the consensus accuracy I talked about, and this is the depth. So if you put 20 strands together as the depth, you can hit about Q40, which is a 99.99 accuracy. This is with our current most widely used chemistry, but I will tell you later that we are improving these things further. So one of the reasons the accuracy was a problem with nanopore sequencing is the homopolymer problem. So homopolymer is because of the architecture of the nanopore. When a DNA strand is passing through, the region that it reads the DNA is what we call kind of narrowest region, is called the reader head. So at the moment, if more than 5 base, so when the DNA strand is passing through, about 5 bases dominate the signal. So if the DNA is a mixed strand of like AGCT, then the accuracy is absolutely fine. But when there are regions of homopolymers, whether it is poly T or poly A, poly G, so you can see the signal is a little bit flatter, which means it will has less features to identify how many bases are there in this particular region. So which means the accuracy is slightly lower because you lose some of the at the end or at the beginning. So you lose bases. So for example, if there are like 10 T's in one stretch, you might call that as 9 or 8 rather than 10. So we are now working to improve these homopolymer issues. To do that, we have found a new pore, which is called R10 series. So in R10, we have elongated the reader head, or we put a separate reader head, which we call the double reader head. And by doing so, we kind of make it less flat. So you can see compared to R9 versus R10, the poly T region it has more features, which means the accuracy improves. So as you can see here, so when you do the pileups in the R9 series, you can see these are the errors. So instead of calling this all poly A at the beginning, it misses some bases. So in R10 series, you can see errors are much less, which means the accuracy is improving. So with this R10 pore, we have improved the accuracy, and we are just launching this R10 series. And with that, we have passed Q20, which means 99.9% accuracy. So with the depth also, like you can see consensus accuracy, this is R9 and this is R10. To the depth requires to reach, for example, Q40, Q50 is now much lower than in the R10, R9 series. So accuracy of Nanopore is keep improving. We are still a young technology, and this level of accuracy is plenty for many applications. And people are happily using Oxford Nanopore Technologies for this. So we have also people asking, so it takes some time sometimes to get these very high accuracies. Informatically, it takes time to read. So if people want very fast answer but less accuracy, so we have different models to be used that you can use to base poll, and you can just get the initial initial impression of the answer that you are looking to the question that you are looking answers for, and then you can, if you want a better accuracy, you can read the same data with better base caller, which will give high accuracy. So depending on, that's a very kind of versatile system. So depending on your requirements, there are many different algorithms and models that you can use to base call your data. We also try to use improve accuracy even further. So I was explaining earlier that we use one strand of DNA to let it pass through the pore to read as the sequence. We are also developing methods to read both strands when the second strand goes away. So we have methods to capture that as well, and then you can combine data from both strands, which will improve accuracy significantly. And with this duplex accuracy, we are hitting Q30 already. So we are still working on to improve and launch this as a product. So this is basically nanopore sequencing chemistry and accuracy, and so you can use these nanopore systems in many different applications. So why nanopore sequencing is important? Because as I said earlier, our motto is sequence anything, anyone, anywhere. Workflows are very simple, and library preparations are simple as well. It takes much less time compared to some of the other technologies. And also there is no capital cost. The midline is only $1,000. But if you want to get larger platforms, how the costing structure works is there is no capital cost, and the cost will come from reagents and other later on add-ons and so on. And Nanopro is the only portable system, and also its data is real time and on demand. So data, while you are running the experiment, you can get the answer you are looking for, and you can start and stop as required. You don't have to run for hours and hours and hours just to get the experiments completed to get the answer. And also it's very information rich, high data volumes, and no you can read small DNA fragments as well if you want to, but you can read very long ones as well. Most of the other technologies, either it's small or long, and Nanopro is the only technology that you can use both long and short reads. And also one of the most important thing of Nanopro sequencing is Nanopro is the only technology that can use for direct RNA sequencing without doing cDNA conversion, which means you can keep all the modifications and other information that you might lose when you do PCR to convert RNA into cDNA. So these machines and platforms can be used in people been using them in many different applications: outbreaks and pathogens, human genetics, transcriptome analysis, environmental analysis, plant, animal genetics, and so on. So I'll just try to give a few examples of some of the key things that over the years people have used this technology for. In agriculture, for example, this is cassava plantations in sub-Saharan Africa, which is many of. And apparently, there are 800 million people rely on cassava, not only as their main income, but also their main source of food. There was a infection that killed entire plantations of cassava. So team from Oxford Nanopore and some of our collaborators, when they sequenced these cassava plantations in the field and identified exactly what was the problem, what was the fungus, and then they managed to get those fungus resistance versions. And the next year, apparently, the yield was much higher than in any other years. So in Sri Lanka, so there are a lot of, Sri Lanka is an agriculture-based economy, so there are a lot of things that you can do in using sequencing. So that's something that I wanted to work with Sri Lankan academic researchers, so you can understand whether is this the plant breed I want, whether the microbiome, what microbiomes in the soil, is this good for a farmer? Is the plant is diseased? And if it is diseased, what is the what best version or variety that is resistant for some of these diseases and so on? And also in aquaculture, it is big. People are using this to monitor waters to see which algal is blooming and whether it's threatening the fish stocks and so on. So Pradeepa is actually trying to get Oxford Nanopore sequencing technology set up in Agricultural Biotechnology Society Center. So she's going to do RNA sequencing of tomato, and she also wants to do microbiome metagenomics of compost and so on. So it's really nice to see that DNA sequencing, Nanopore sequencing technology is being kind of established to understand some of these problems. So you don't have, as Pradeepa said, you have to otherwise send samples to USA or Korea, but with Oxford Nanopore systems, which is very cost effective, portable, so you don't have much lab space, you don't need much lab space, and so you can develop these systems in-house and get the answers you want. So one of the other application is sort of like antimicrobial resistance. So like it's becoming big and big and big. So there are reports saying that by 2050, antimicrobial resistance will kill more people than cancer does. So it's very, very important to understand these systems and how to get around these systems. And I'm really pleased to see that R&D Jayasinghe from Botany Department, Faculty of Science of Peradeniya, is also trying to establish nanopore sequencing technology to understand antimicrobial resistance and so on. It's also big in food supply chain monitoring. So if you take a burger, for example, when it comes from the farm, from the cow to the consumer, there are many different stages in the supply chain. So if there is a contamination, if you monitor these stages, you don't have to call the entire production lines if there's a contamination. So most of the big food companies are now trying to establish DNA sequencing technologies to monitor the supply chain. So it saves billions and billions of dollars, so that they don't have to recall the entire production lines. It's big in cancer research. So in cancer, like obviously, it's a DNA variation, structural variations, large insertions, deletions, translocations, and so on. Longer DNA read sequencing methods are much better to understand these structural variants compared to short DNA sequencing technology. So as a result, nanopore sequencing is people are using all over the world for many different applications of cancer research. And this is the only sequencing technology that NASA took to the space, to the International Space Station. So in the first picture, you can see the midline is plastered around the boards, and then they were doing sequencing in space. So this is to monitor for various objectives in this project. Like one is to monitor the health of astronauts. At the moment, they have to explain their symptoms to doctors on the Earth if they get ill. But if you can sequence your own saliva sample, you can tell them, I have exactly this infection, this particular virus or bacteria. So you can get very specific medication rather than trying to do trial and error treatments. And also they monitor the International Space Station with swabs, and they obviously, in case if there is extraterrestrial DNA, so you can sequence. And they took it to the there's another NASA lab in the deep sea labs, and they took them to these deep sea labs as well to do many different applications. And we are working closely with NASA to in the exploration of Moon and Mars and beyond, and is quite nice collaboration. So in the future, like imagine you go to you go to a doctor, and if every doctor's office has a MinION, and they can take a swab and they can let it sequence while the patient explains the symptoms, and hopefully within a few minutes they will know exactly which bacteria or viral infection people have. And with this MidGiant, for example, the mobile unit, people can do it even at home, and you can go to the doctor with that information in hand, and real-time monitoring and track your health. So at the moment, people are using quite a lot of devices to monitor their health, you know, heart rate and so on, but you don't really monitor your the factors in the blood or in your systems. There are thousands of them that you can monitor, but in the future, if there are portable systems that you can monitor, so we will be in a different space in terms of monitoring health. So all these things are possible with portable sequencing or sensing technologies. We also have this program called OB1, and this is to support biodiversity to existing customers. Nanopore sequencing customers, we provide reagents free to sequence IUCN red listed species. So I'm pleased to see some of the labs in Sri Lanka actually are in discussion with us to use this program to sequence some of the endangered Sri Lankan species as well. So nanopores in outbreaks. So obviously, because of COVID, I thought I'll talk a little bit more about outbreak situations. So the Oxford Nanopore systems are ideal in these kind of situations, mainly because it's portable, it's easy to distribute, and it's near sample testing. And also the power requirements are low. You can set it up anywhere. and ease of use and rapid answers. So people have been using Nanopore systems to monitor outbreaks over the years, and this is one example of Ebola outbreak when it happened in Africa. A team from Birmingham University actually went there with Nanopore systems in their luggage, and then they set up makeshift labs and they started monitoring samples. And they say that this is a paper from Nature. This is how they kind of try to sequence samples in these makeshift labs. And it took only like 15 to 60 minutes for them to sequence DNA and identify Ebola. So it's been used in later outbreaks of Ebola as well. And so you can see, like, you can understand which variant is spreading in which region. And this is key information to control outbreaks, and it gives policymakers the power to take right decisions to control these outbreaks, to make it not become a pandemic. This is also for yellow fever, a similar situation. People have used nanopore sequencing. In coronavirus outbreak, so when this happened, Nanopore acted very quickly to develop systems. So when with coronavirus, so you will have clinical signs and symptoms, whether the temperature or cough. So you won't answer whether the patient is now having COVID. So then you do the diagnostic confirmation using PCR. So it will tell you whether you have COVID or not. But then what is important is the genomic epidemiology, which is the sequencing information that provides you, provides especially the health systems and policymakers. This is the variant that is spreading, and then they can take measures to control the outbreak. So we acted very quickly when COVID happened. It started in obviously in Wuhan. So the Chinese CDC lab very quickly moved to Oxford Nanopore systems, and then it spread all across the Chinese CDCs, and then went to America, and they all across Europe as well. So this is the SARS-CoV-2 genome, and the critical part of the spike protein is this bit of the gene, and this is where most of the vaccines development is targeting this spike protein as well. So to understand whether any mutations happening in these regions, especially key regions, and whether they are responding to vaccines or not is key to control the pandemic. So Oxford Nanopore technology is being used in more than 100 countries in these kind of studies. So we have developed protocols very easy to use, and also we package all the third-party reagents together as well. So when people buy COVID packages, they just have to buy ONT package. They don't need anything else, and then you can get answers very, very quickly. So in Sri Lanka, when COVID happened, Professor Nilika's lab in Japura University was the only lab that was properly established to do genomic sequencing of COVID. They were using a different method, but that was very costly, and as a result, their capacity was very, very low. They could only do less than 100 samples a month, because each sample apparently cost about like 50,000 rupees. So when I got to know this situation, I kind of talked to my CEO and donated Nanopore technology systems to Nilika's lab, and they very quickly, within weeks, they were up and running with Nanopore systems, and the capacity improved significantly. So with Nanopore systems, you can do, if you want to, you can do like 96 samples a day rather than doing 100 samples a month. So they were very happy with the systems, and recently they published a paper comparing Illumina versus Nanopore, and they are talking about how effective Nanopore system is, how easy to use. Sample preparation time is half of the Illumina technology, And then the cost is only a fraction of Illumina as well. So they were very happy. And then other labs slowly started doing nanopore sequencing as well. And Professor Enoch Pereira's lab in University of Columbus Science faculty started doing collaborations, and they set up sequencing facilities. And then it slowly spreaded into government labs as well. Ministry of Health got involved through UN funding. They set up 4 labs, 2 labs in Medical Research Institute headed by Dr. Nadika Janage and Dr. Janaki Abhinayaka. And then there's a lab in Kandy Hospital Virology Lab, Dr. Rohit Abduggal. I was just there in the morning. It's really nice to see, like, it's a very kind of tiny lab, and you can do sequencing only if the machine is tiny. So the nanopore sequencing is ideal for that kind of setup, easy to use, portable, and you get real-time data. And the other one is in Karapati Hospital. So more labs will be set up soon. And Professor Fasih Hanuddin in the Peradeniya Medical Faculty wants to set up COVID sequencing facility in Peradeniya Medical Faculty as well. So this is actually the report came from Professor Nelika's lab on 31st of December last year, just to show that which variants are actually spreading. And out of the 176 samples, they sequenced, there were 41 Omicron samples, and this information where this in which region, which species is, which variant is spreading is quite important to combat pandemics and to make right policies to take remedial measures. So as I said, our motto is to sequence anything, anywhere, by anyone, and it's already being done. So people do sequence in any environment, in the Arctic, in the Antarctic, in jungles, in the field, and in the space, and deep down the sea. So we have actually achieved our motto, but like we will improve things further. We will make it more accessible, cheaper, more accurate, and make it more high throughput. So nanopore sequencing will be hopefully household name. So it's nice to see that in Sri Lanka also now finally nanopore sequencing is now being established in many different labs. So this is my contact details. If you have any questions, please let me know. Like if you need nanopore systems, please let me know. And we have a fantastic technical support team and customer support team in Singapore. They have now very dedicated team to help Sri Lankan customers. So I'm really happy to see how everything planned out over the last 6-7 months. And yes, so let me know if you have any questions. So I think that's it.
Thank you very much, Lakmal, Dr. Lakmal. Any question from the audience? I see one question. If we go for this technology for DNA sequencing, what will be the cost per sample? I guess it depends, but you can.
Yeah, so it's really depending on the what you are sequencing. For COVID, for example, if you do, so you can do either one sample, or you can do like 96 samples in one go. So if you do 96 samples, so the cost goes down significantly. So you can do 2048 samples in a larger formats. So then cost goes down to less than $10 a sample. And even if you run 96 samples, cost is about, I think, $15 per sample, which is a fraction compared to some of the other technologies. But it's really depending on your application, which system you are using, what answers you are looking for.
Anything else? You can unmute and ask the question, or you can type. Okay, so there's another question. Is flow cells reusable? How many times?
Flow cells are depending on the applications. We do give a certain time frame for a flow cell. We say, for example, because flow cells are there's a lot of chemistry inside the flow cell. There's a lot of like mediators, buffers, and when you run the experiment, they come out. So if we say, like, say flow cell is, you can run continuously, like say for example 48 hours. But if your experiment takes only 10 minutes, then you can stop in 10 minutes and then you can run again, and you can sort of combine time, could be the time that so you can reuse for many times, again depending on what is the application and how long you want to run it for to get the data you need. I hope that makes sense.
Yeah, I hope the answer is there. And again, there's a question: is it possible to sequence plant RNA for transcriptome assembling using Amino?
Yeah, so the transcriptome analysis is big with Nanopore technology. Actually, we've been working with the Oxford vaccine group that developed the AstraZeneca vaccine. So they were using our system. They use Illumina as well, but they were comparing, and they also published a paper saying how nice to work with Nanopore technology to do the transcriptome analysis. They were checking how when the vaccine is given, how the transcriptome changes and so on. So transcriptome analysis, yes, you can do plant genomes and so on with me. Depending on the, again, answer, the platform you need to use can be changed. If it is a small problem that you're trying to solve, it is a small genome, then you can use a Min9. If you need much high throughput, then you have to go for the other systems. But even with Min9, you can get about 50 GB of data in one flow cell, up to 50 GB.
Thank you. There are so many questions coming up. Is the technology approved for clinical use?
It's really depending on which clinical use. Again, I think for COVID, it is being approved in Europe. In Sri Lanka, I'm not sure how things work, the system work, but for diagnostics, if people want to use, I was told by Nilika, for example, for all the COVID work, they didn't have to get any approval. Everything is fine. But if it is required, I was told that it's not that difficult to get the approval.
Okay, it is answered. And then there's another question. Is there any special way to prepare samples for this sequencing?
There are many different ways of, again, depending on the application, depending on which platform. So we have fast sample prep methods that takes only like 5-10 minutes. But in that one, yield could be lower. And if you want high yield, you can spend a little bit more time in sample prep. Depending on the DNA, it also affects the yield. So it's really depending on the problem again. But yeah, so many options are there for sample preparation. All the kits are there. It could be the manual or automated, but it's not that hard at all.
This question is also related to that. A sample itself for extracted DNA should use.
At the moment, it is extracted DNA, but we are developing sort of like liquid biopsy sort of device that people can take, for example, saliva sample, just put into a tube, and then sort of injection or syringe sort of thing, and then you press the syringe and deliver the sample directly to the Min9. So that is in development. But at the moment, yes, you need extracted DNA or RNA.
There's another question. I will answer the next one since it is related to AGBC. So I'll keep that. Usually, when longer read sequencing is CAM, DNA quality is a big issue. DIN number and amount needs to be high, and usually we fail in that aspect, especially when it comes for PacBio. What are your thoughts on this aspect when it comes for Nanopore?
Yes, as I said, I think when in Nanopore, when there's no size limit, whatever you can extract, you can put into the system. It will read. It will be read. True, like when the DNA is longer, then you will have other issues like DNA may be in different 3D structures and so on. But again, that depending on the DNA, that problem varies. For example, if you have like chicken DNA, for example, apparently notoriously difficult to work with long DNA, but some of the others are fine. It really depending on the system, but on average, I think it's depending on also how long. Rather than having few hundred, and you can easily have like 5,000, 10,000 long fragments without much issues at all.
Okay, it's answered. I hope, yeah, she wants to thank you for the nice talk as well. And there's another question. Is it really possible to get 50 GB data per flow cell, or is there a drop? If yes, how to eliminate?
So there are, again, as I said, like some, depending on the DNA sample prep, and the type of DNA you use. And in normal, like so bacteria, sort of easy to work with DNA, you can hit 50 GB, but I mean, that is kind of maximum limit. But if you do the sample prep right, and if the DNA is not problematic, you can get those sort of numbers, yes.
I think this is also related to that. Can we use low quality degraded DNA for analysis?
I think you can, but again, then the yield will be low. But if that is the sample that you have, then you have to obviously work with it, but then you have to accept the fact that you may not get high yield.
Okay, this question, if I wish to establish this technology in my lab, what would be the cost, Minion? And contact point for Tamil Nadu, India. So participant, he or she is not from Sri Lanka.
Okay, so we have actually a sales and marketing team in India. So I can, if you email me, I will provide you the details of the Indian team. And the sequencing cost, if you want a MinION, it starts with $1,000. For the $1,000, you get the MinION 2 flow cells and one sequencing kit. So that's the initial setup, and then you have to buy the reagents and more flow cells and so on. But initial setup is that, and then you will. There are a few different packages, and you can get I think something like 3,200 or so. You can get a slightly bigger package with MinION, I think a few more flow cells and maybe 2 kits rather than 1, and so. So all the details are on our website, and there are many different options available. But it's not, you don't have to rob a bank to set up Nanopro sequencing facility.
There's another question. Is it possible to sequence a fragment of very low number of base pairs, 40 to 50 base pairs using Nanopore technology? Would,
yeah, I think if it is longer than the piece that is inside the enzyme and the pore, should be okay. I think 50 should be fine. Yeah.
Since this question is related to AGBC, I think I can also help with that. Do you provide this Nanopore sequencing service via Agricultural Biotechnology Center, Perabenia? If so, if the sequence service is done by you, whether the analysis is also done. Yes, actually, we are trying to establish this system. I mean, you know how hard to get things down to Sri Lanka. So anyway, we are almost there. So yes, we are planning to become a service provider for this service. And if so, we will also have an option for the analysis as well. We can, I mean, even in foreign labs, they do those two separately. Either you can get the wet lab or the sequencing part done and analysis by yourself. That's what we have been doing. Since we have a good bioinformatics facility, I mean, we have set up those things already. Therefore, we can provide the full service as well if necessary. I think I answered that question. And is it possible to see? Yes. How much will it cost? Well, I think we
answered that question. I think it's very depending.
Yes. Yeah. Yes. Anything else before we wind up? No more questions? Okay. Yeah. I think we had about 100 in the audience, and I think it's a very good discussion as well as a talk. Many of you mentioned that as well. So I take this opportunity to thank our speaker, Dr. Lakmal Jayasinghe, once again. Thank you very much for agreeing to give this talk and helping us to establish this system in Sri Lanka. So yes, we will need your support in the future as well. It is a really good collaboration, I guess. And I want to support, like I said, it was a dream. When I took the directorship at the Agricultural Biotechnology Center, one of the dreams was to establish a sequencing facility in Sri Lanka. So it's becoming kind of true. We'll see how it goes in next few months. So yes, then if we get it done, we will be able to provide the service to all the Sri Lankan scientists as well. Obviously, there will be a cost, but will be less hassle for sure. You don't have to get all the permissions from here and there to send your samples. Therefore, it will be less time consuming and obviously cheaper as well. We will just do it for the cost. Agricultural Biotechnology Center, not a business organization or a business body. So we are just a university service provider. So we will try our best to minimize the cost. If Lakmal and others support, we can do that as well. So thank you very much once again, Lakmal. We are very proud of you becoming a top management in a big global company. So thank you very much for being here and helping us to understand this technology. And I take this opportunity to thank all the members who joined with us. I saw a lot of people from other universities and most of the known names, obviously. Sri Lanka is a small country, Department of Agriculture, other research institutions, many other universities from Sri Lanka as well. So we had a really good participation, and all the students I hope you also got an idea about this new technology. Thank you very much once again, and we will keep in touch. We will update you. Once we establish the system, we will definitely pass the message to all of you. Thank you very much, and we will wind up here. Thank you.
Thank you. It's a pleasure to be here. I can end meeting for all, shall I?